Dr. Kar. I think to introduce Dr. Kar. She should have done. Yeah, to introduce Dr. Kar, I think she has been my colleague when I was in NRRI Cuttack. Of course, that time it was known as CRRI Cuttack. She has been very diligent researcher, and I have seen her growing in her academic achievements throughout her life. She is the principal scientist at the Crop Improvement Division of the ICAR National Rice Research Institute, Cuttack, Odisha. And she has an amazing devotion towards her work. She received the Women Inspiration Award from Lions Club, setting an example for the upcoming youth. So, thank you, Dr. Kar. Maybe you can just.
Thank you, sir. Respected chair, co-chair, esteemed delegates, ladies and gentlemen, many students are there. So today's lecture is fusion of genomics and conventional breeding strategies towards breaking yield barrier of indica rice. Just before me, Dr. Jena has elaborately told how he has utilized his knowledge and in enhancing the gene pool so that he could derive many useful genes from the wild background. And now we'll discuss something, how we'll, because through conventional breeding methods, we have come up to a great extent and we have achieved green revolution through this. But now a time has come, along with the conventional method, we have to integrate some other cutting edge technologies so that we can break the yield ceiling to feed the burgeoning population. So rice was domesticated some 7,000 years back, and the farmers, when they started domesticating, the first farmers perhaps, that time only unconsciously they started breeding. They started selecting the lines, the materials which are good for them, either from taste point of view, yield point of view. So breeding started at that time. But for a very long period, it was just like only through farmer selection like that. So if you can see, farmers were perhaps they were the first breeders because they were much more concerned about the yield potential, about the quality preference, about disease, pest, reaction. So they started consciously, they started selecting. But there was no hybridization program at that time. Only selection was going on, and many land races, you can see, many land races were developed by farmers. Over the years of cultivation led to development of many land races, and those were many popular land races I have shown here. But besides that, thousands of land races were there at that time. Perhaps now most of them are in the gene bank because they are now no longer cultivation in the cultivation form. But still we are growing Gobind Bhog, Kala Jeera, some scented varieties. Still they are popular with farmers because of their very unique cooking qualities. So after that, when the land races were already well developed, but they are having some inherent problems. Their yield level is very low. They are prone to lodging. They are tall plantains, very few tillers. So actually our organized rice breeding program started in 1911 by Dr. J.P. Hector, the then economic botanist, and the first research station was established in Dhaka. Then subsequently, emphasis was given, and by 1950, 82 research stations in 14 states of the country were established. And these research stations, they were mostly from 1911 to 1949. The focus was mostly on pure line selection, because we all know that land races are a mixture of pure lines. They were maintained by our ancestors for years together. So our scientists, they started selecting those land races, they started purifying. And many good pure line selections were developed, and even some of them were very popular for a great extent and for a long period of time, and subsequently they were used in the breeding program. And if you can see in our state, in Odisha itself, there were 3 stations, Cuttack, Barampur and Jaipur, and they started purifying the materials available. Cuttack started T series, Barampur BAM series, and Jaipur J series. And many lines you can see T141, T90, T1242, BAM 6, SR26B, FR13A, where from we got the sub 1. And those materials were developed that time. And but they also carried the same legacy. They were also very low yielders, and only they were purified form. So there was a great pressure on the scientists to increase the production. And after the Second World War, towards 1949-50s, there was a pressure for increasing the yield by applying fertilizer, chemical fertilizer. But unfortunately, all these pure line selections, as they were tall, they were prone to lodging. So after application of fertilizer, their condition deteriorated. So they all, there was great lodging and yield was low. So that time only, the typical, what we tell, by that time also Mendel's laws of inheritance and scientific breeding procedures, already they were well known. So that concept of hybridization and development of hybridization based varieties started in 1950s by the establishment of International Rice Commission and our institute CRRI, and at presently it is named as NRRI. They were the pioneers, and the war started in 1950 to 54. So through indica japonica interracial hybridization, because japonica varieties were dwarf types and they were very much responsive to fertilizer. So keeping that in mind, that interracial hybridization program started. But that program didn't result into good materials. However, 3-4 varieties were developed that time. Melingia, Masuri in Malaysia, ADT 27 and Cisna in Australia. Though out of that, Masuri was cultivated for a long extent and it was very popular with the farmers, and that was subsequently used in the development of many mega varieties. The present day mega variety Swarna is also a derivative of Masuri. So, but the great impetus we got when towards 1965, that Taichung native 1, which is a derivative of Digio Ugen. So that dwarfing gene ST1, that concept started, and then hybridization was done with the local indica varieties and the typical semi-dwarf Taiwanese varieties, TN1. So our first varieties, semi-dwarf high-yielding varieties, Padma and Jaya, they were released in 1968 out of the cross between T141 and TN1. And the typical conventional breeding started. So after that, so many, now more than 1,000 varieties perhaps they are released through conventional breeding technology. So out of that, you can name Miracle Rice IR8, which perhaps in 60s and early 70s, that changed the scenario in rice cultivation. So yield was double, triple in many instances. They were semi-dwarf varieties. Out of that, we have till now so many varieties are very popular. The Swarna, Samba Masuri, M2 1010, M2 1156. Our very progressive farmers, he was telling about CR 1009. They were still now, they were developed through conventional breeding and they are very popular with the farmers. Then premium basmati varieties, Pusa Basmati 1, Pusa 1121, and our first high protein variety, CR 310, they were all developed through conventional breeding method only. But conventional breeding is having its own limitations. It is a very time consuming process. It takes almost 10 years for releasing a variety. And also, we do not know what genes we are transferring. Only it is a number game. Because once you will make a cross, if the character is a very complicated trait, then you have to grow a very large population. Out of that, you may get a good variety, a good line. You may select a good line. So And also after 2 decades of cultivation of all these semi-dwarf varieties, and it was noticed that there is no longer increase in yield, there is a plateauing of yield. So scientists started thinking how to break that yield ceiling. So different technologies available, there were different concepts were utilized in rice breeding also. So out of that, one is idiotype breeding. Though the idiotype concept was very old, it was given by Donald in 1968. But when there was a yield plateauing, so scientists from ICAR-NRRI, particularly Dr. Fuchs and his group, they started thinking how to model. We have to go for modeling of rice plants. So different traits they tried to combine, because earlier indica varieties, tall indica varieties were side tillering types. But in between we got very high yielding varieties with large number of tillers. But out of that, many were unproductive tillers also. So to counter that, they started thinking if we can develop a variety with low tillering capacity, with high grain number, then with a plant height 90 to 100 centimeter, with thick sturdy stem and dark green leaves, vigorous root system, then certainly we can increase the yield. So this led to development of first generation of NPTs. That time, bulu varieties, means tropical Japonica varieties from Indonesia, they are very vigorous type and they are having very good traits. So those lines were combined, but unfortunately that program failed because the yield level was not that high, mostly because of the low biomass and poor grain filling. So again, scientists started thinking of developing second generation of NPTs, using those first generation of NPTs and crossing them in the breeding program with indica varieties. So in our institute, in National Rice Research Institute, we have also developed so many, we are telling new generation rice varieties, keeping that concept in mind. Recently we have released last year Siadhan 316 in one variety, which is having very heavy panicles of 6 to 8 gram average weight, high grain number with good quality grains. Tillering is less, 6 to 8, 7 tillers. But we are focusing on increasing the biomass. That's why we are giving emphasis on intermediate height, 115 to 20 centimeter height. And top 3 leaves, they are the major contributors of photosynthesis, that long semi-red top 3 leaves and strong thick columns. And you can see the panicles, how big they are. Even the average grain number I have shown 250 to 300, but some of the panicles are having more than 500 grains also. So now we have, this is through conventional crushing only, but developing, but using a diverse source of materials, starting from tropical Japonicas, Indicas, different new generation NPTs, so many materials our scientists, they have utilized for developing all these new generation rice varieties. For breaking, and our purpose is to break the yield ceiling. However, there are also some inherent problems also, as their growth is very good. They are also equally susceptible to many diseases and insect pests. And for stabilizing those problems, for biotic stresses and abiotic stresses, we have to incorporate different genes which are already available, and we are doing that. Particularly, these lines are highly susceptible to BLB, sheath blight, and we are addressing those problems. So then another concept also started coming. We have to heterosis breeding. It means hybrid varieties, large number of hybrid varieties are developed, and yes, sir. I'll complete. And here many of our partners, means they are from seed industry, and more than 20-30 varieties were released, hybrid varieties were released. But they are also having some problems, because that seed cost is high, and every year you have to purchase seeds. So they are not, means though they are having more than 15 to 20% high yield, but still they are not that much. Area expansion is bit less, and but this is a good technology. This can certainly break the yield ceiling in near future also. So another concept came, Sar has already given in detail, marker assisted selection. Because before that, we do not know, we didn't know that which genetic segment, genomic segment we are transferring to our variety. But as the markers were developed, molecular markers were developed, so our scientists, our breeders, they started using this already well-known clone genes or gene-based markers or tightly linked markers in the breeding program. And marker-assisted selection is no longer, it's almost a conventional method now. And in our breeding program, we are routinely using, but they are having their own problems also. You can see, so far more than 30 varieties we have released in our country through marker-assisted selection. But you can see the major focus is on only biotic and abiotic stresses. But nowhere we have simply bred for yielding high yielding varieties, because yield is a very complex trait, and marker-assisted selection is not working very well. Simply, if you will transport two to three genes at QTLs, then you may not achieve that much level of high yield. So that is the major limitation there. So now another approach is coming, that is the extension of marker-assisted selection, genomic selection. Because now so many cutting-edge technologies are available with us. Now the sequencing cost has come down. We can go GBS technology, genotyping by sequencing. This is available. Large number of markers that markers are high density genomoid markers are available with us, and then sequencing platforms are there. So now we can use all these things in going for genomic selection, so that we can address all these complex traits like yield. So here our target should be, we have to exploit the novel variation in the germplasm, accelerating the breeding cycle, that is speed breeding, then high throughput precise phenotyping and genotyping we have to do. We have to, means beforehand, we have to know the genomic estimated breeding value, so that selection will be easier. Then already allele mining, gene cloning, all these things, they can add up to our genomic assisted breeding. Instead of marker assisted breeding, markers, we can go for genomic assisted breeding. Then, and also, though it is in a nascent form in our country right now, but progress is already there. So many platform projects are coming by CJR institutes and ICAR Bill and Melinda Gates Foundation. They are focusing on next generation breeding, genotyping, and digitization approaches. And our institutes are all a part of it. So, and this is just a flow chart, how we can use our knowledge, scientific knowledge from different sources in genomic selection process, but it needs high throughput genotyping and phenotyping platforms are also now well developed. So we can use all these cutting edge technologies in our breeding programs, and our breeders are, we cannot do everything, and biotechnologists, they are there to support us. And this is our plant genomic facility developed at, in ICAR-NRRI New Delhi, that high throughput phenotyping we can do. Then this is, we have to integrate everything. Even the conventional method, though they are very time consuming and they are having their own problems, but they are the time tested ones. So they should be integrated with different cutting edge technologies, so that we can assemble. Now breeding is, we have already started with Dr. Jena and the ICAR group in developing genomic assisted breeding for increasing yield potential also. There we targeted GN1A, SPL14, and we transferred it to two institutes, ourselves from NRRI and our IIRR partner, where we are a part of it, and we developed breeding lines in 3 mega varieties, Swarna, Samba Masuri, and MTU1010, by incorporating different genes, GN1A, SPL14, and you can see the MTU1010 panicle and our improved lines with all these genes and QTLs. Our Hyderabad partner, they have extensively studied them, and some of the lines are even more than double in yield. So you can see the plant architecture. So So rice breeding, actually the conventional breeding is a breeding by chance. Whether even if we ask the breeder who has developed Swarna variety, sir, whether you can develop from same Basista and Masuri cross Swarna line, but he told may not be, because we didn't know which genes were combined in which segmental regions came from what. But now, as the science has progressed a lot, now we can go for breeding by design. We can design our own crop, design our own varieties. And so certainly it should be amalgamation of all the technologies right now, whatever is available, so that we can yield, we can expect that we'll get a rice variety, which will be more than now our target is 10 ton, which through all these super rice and all these NGR varieties we can achieve. We can go beyond that, even we can target for 15 tons, and we can feed the burgeoning population. With this, so I'm concluding my talk. Thank you. Thank you, the organizers, for giving me the opportunity to speak a few lines before you. Thank you.
Thank you, Dr. Kaur, for the nice presentation. I think, yes, plant breeding has come a long way from pure line selection to speed breeding. And then she has nicely collected all the information. I think for the sake of brevity, we will wait for questioning, and then we will go for lunch. Maybe we can interact there. I think that will be much better. We are already half an hour late, so I don't think that food should wait for us. We are working all these things for food. So now 1:30, it's now 1:30. We come back at 2:30 and then finish up the rest 2 lectures. In that case, probably we'll be able to save time for the next session. Thank you very much.